As we all know, dendritic cell is actually activate any naive T cells. This could be T helper cell or cytotoxic T cells. So cytotoxic T cells, when gets activated, it is naive in conditions. When get activated, this cells is called cytotoxic T lymphocyte. This means activated, active T cells, cytotoxic T cells. The main function of these cells, one is it kill virus infected cell directly, and the second is kill tumor cell, that is also directly. So these are two functions performed by this cytotoxic T lymphocytes. But how these cytotoxic T cells activated? Because it was at first in naive conditions, inactive in conditions, then it becomes cytotoxic T lymphocyte, CTL. And as we know, all naive cells are activated, all naive T cells are activated by dendritic cells. So dendritic cell is responsible for these activations. But we know that dendritic cell is called professional antigen presenting cells, and we all know that professional antigen presenting cell is actually show MHC2 molecule specifically, because all nucleated cells have MHC1 molecule, and the cytotoxic T cells actually interact with MHC1 molecule. But for its activations, no other cell can activate, because other cells like muscle cells, tissue or other organ cells, like I want to tell you that a virus infected cells cannot actually activate in naive cytotoxic T cells, because no other cells have a co-stimulatory molecule that is called CD80 and CD86. So CD80 or CD86 or both, these two molecules is very important for the activation of any T cells. It could be T helper cell or cytotoxic T cells. It is very important. But here is a problem. Dendritic cells much of the time show actually by MHC2 molecule, because it is professional antigen presenting cells, and professional antigen presenting cells present antigen by the help of MHC2 molecule. But for the activation of cytotoxic T cells, MHC1 molecule is important. As we know that dendritic cell have nucleus, so dendritic cell can produce MHC1 molecule. But how these dendritic cells get the virus antigen or the tumor antigen? So for that reason, a very important thing occurs, I want to mention here, that's called cross presentation, or you can say it is cross priming. This thing actually occurs. This is very important. Same thing, cross presentation and cross priming. What happens? These dendritic cells engulf this virus infected and tumor cells. So what happens is dendritic cell actually engulf these virus infected cells and tumor cells and get the antigen and represent the antigen by class 1 MHC molecule. That is called cross presentations. That is not actually generally occur, but some dendritic cells are capable of to present the antigen through the class 1 MHC molecule. So this is very important for TC cells activation. So what happens? Now these dendritic cells express or actually present the virus antigen or the tumor antigen to the TC cells that is naive in conditions, and these naive TC cells become cytotoxic T lymphocytes that is activated. Now these cytotoxic T cells can perform its killing functions by, first is it secrete perforin, a protein that actually form pore in the plasma membrane of these virally infected cells and tumor cells, form pore like structure, same like the C9 molecules of MAC, means membrane attack complex of the complement pathway, form a pore like structure. So perforin form a pore in the plasma membrane of these infected cells or tumor cells. Another thing is granzyme. It can secrete granzyme, and granzyme is actually serine protease that actually helps in apoptosis. I am not mentioning here details of the apoptosis. Actually, granzyme actually cleave a protein called bead. So I am not talking about in more details, but granzyme actually helps in apoptosis. So the cell die by the granzyme. There is another chemical called granulysin. It digests the cell, granulysin. The fourth thing is this cytotoxic T lymphocyte express a very important molecule called Fas ligand. Fas ligand. That Fas ligand can bind with a molecule called Fas. That Fas is actually presented by these virus infected cells and the tumor cells. So viral infected cells or tumor cells have Fas, and they interact with Fas ligand and apoptosis occurs. Not only that, there are many other factors are present by which the cytotoxic T cell can directly kill a cell. That is lymphotoxin alpha. This is a cytokine, or it has another, it is called TNF beta, and it can secrete IFN gamma, TNF alpha. So by this many way, the cytotoxic T cells can kill the virally infected cells and the tumor cells. Okay, so after it gets activated, the naive TC cells become cytotoxic lymphocytes. Then these cells actually reach to the virally infected tissue site or tumor site and can kill these cells. And there are two subtypes of CTLs. So CTL can be divided into two subtypes. CTL, two subtypes, that is CT cytotoxic TC1 and TC2. There is no very diversity in this subset, because in case of T helper cell we have seen that TH1, TH2, TH17, T follicular helper cell, T Treg, and there are other subsets of these cells also present. But in cytotoxic T cells, two subsets are actually present, that is TC1, TC2. And very interesting thing is that cytotoxic T cell 1 actually perform like TH1 cells. It can secrete IFN gamma. And another interesting fact is this TC2 can secrete IL-4. So it actually acts like TH1 and TH2, but they are not actually really TH1 and TH2, but they secrete much more like these cells. Okay, another important thing is that this cytotoxic T cell 1 can kill a cell by perforin and Fas ligand mediated pathway, but these cells can only kill by perforin. So you can remember this.